Thanks a lot for organizing all this. I know how much hard work that has been and still is in lots of ways. And I will try to stay away from the word free will. Especially now with the last question, we've been thinking also, but also before already, been thinking about what is a decision and how to characterize decisions and what do they look like. I'm a neurobiologist, so I'm going to try to present some examples, because in the last 10 or 15 years or so, we've been able to look in great details about how circuits can make decisions. And we've been able to, we've started to be able to do that in animals with fewer neurons, which is where it's more easy to find the circuits that are involved in controlling the behavior. And the examples I'm going to show are by no means the only ones. There's many examples by now, and there's more and more coming. The mammalian people are still quite far away from that level of explanation that we have in simpler animals, but they're catching up. And so it will be interesting to see in maybe 10 years' time, when the first more mechanistic explanations of decisions and behaviors come up, how they relate to what I'm going to show you today. Now, Peter showed, and I hope it was as compelling to you as it was to me, the complexity of what Peter called the neural code and how neurons operate. Now, what he was kind enough for you to not say is that it's a lot more complex than what he showed. So there's not the neural code. We can already see in many other animals that there's many different kinds in which neurons act and operate and interact. So it's probably more appropriate to talk about neural codes. Different brain areas in the human brain use different ways of signaling, and the same is true for other nervous systems. It's also that he showed you a particular type of neuron that looked in a certain way, that operated in a certain way. In the recently completed connectome of flies, our colleagues detected or identified at least 4,000 plus different types of neurons that all behave differently. So not only do we not have neurons that look like a point and a stick, like in AI models. They look all different, they behave all different, they have all different expressions of different genes. So the complexity goes vastly by orders of magnitude and dimensions beyond of what Peter showed. So he was kind enough to give you an introduction into a simplified version, and I'll try to show you a little bit the kind of dimensions that go beyond that. And that's in animals with much, much fewer neurons than we have. So that just to give you a brief glimpse into the complexity that we're facing, and which is one of the bases why I think that people who believe that we need magic to have free will are vastly underestimating the kinds of things that this type of complexity between our ears can actually accomplish, and all the other things that our body has that is not just between our ears. I'm not trying to make too many points. Pick a few points I'm trying to make. One of them is that as I started looking into what a decision is, it turned out that it's hard to pinpoint. And so I chose the examples that I chose to show kind of different settings. And ultimately, this goes back to an old paper about picking and choosing that already, and we'll get to that in a second, that has been seminal in laying the groundwork and saying that, oh no, in some kinds of decisions we have free will, in others we don't. And I think, even though it was never, I think to my memory, made explicit, this has already come up a couple of times also in this conference, is that, well, where do we find the free will? Is it in these decisions or in that decisions? All of this happens in a backdrop where I think Henry has laid out quite clearly that the old notion of asking whether the universe is deterministic or whether it's indeterministic isn't really an old question that has been overcome. What I think has become quite clear over the last 100 years or so is that it's always a fundamentally both in a lot of ways and neither. It's that in some cases, when we choose to see a certain aspect of the universe, it looks deterministic to us. In other places, it looks more stochastic to us. But most of the time for us here on Earth, if we take the perspective of the individual that is supposed to act, to make a decision, to get up in the morning, do all the things that we do, and then go to bed at night, usually that kind of world in which we live is underdetermined. It means that, yes, sometimes it's very clear what we have to do. If I need to catch a train, I need to be at that point in time, at that space. And it is quite clear, very often it is quite clear what I have to do, even though the minutiae of how I do it, cab, walking, public transport, is not fixed. And so it's underdetermined. And also very rarely, things are totally chaotic. Sometimes it may feel that way, but very often it's not. And most of the time we spend our lives in this area. So, and this I would try to argue is also the same, it's also the case for all our ancestors, predecessors, all the way until the first animals crawled around. And so the paper I was referring to about different kinds of decisions that we may have faced over the millennia is by Ullmann Margalit and Morgan Besser, picking and choosing, where the point is, well, one of the points being made is that there are different kinds of decisions. There's some that they call picking, which is where there is no relevance to the two options that one is picking, and then choosing where there has to be some deliberation. And one of the few points I'm trying to make is that there's probably a lot more than just two types of decisions. And so just to get yourself into the mood about what kinds of decisions there could possibly be and what those look like in terms of deterministic, stochastic, or anything in between, I will just show you in quick succession a couple of slides on different types of decisions that I both picked and chose in how to behave and how to make a decision. And I'd like you to, in the back of your head, if you can, some slides I will show just so briefly that you can ask yourselves all of those questions at the same time. It's going to be too fast, but which of those decisions would promote survival? Is this a difficult or easy decision? What would I do if I would be in that decision? Or how much time do I actually have for that decision? So do I have to do this quickly or slowly? Can I deliberate, or do I just have to do it right away, or something bad happens? Right? So reflexes, one can look at that. You know, should I, if I'm that person, move or remain at rest? Do I choose to give my money, or do I choose to give my life? Do I take cover or stand up in such a situation? Do I jump, or do I stay where I am? Do I advance, or do I back off? Should I drink the poison, or rather not? Well, should I buy a Bugatti or a Tata, or should I test drive a Bugatti or a Tata? Should I eat chocolate or broccoli? Should I go skydiving or try to get into space, or live with chimpanzees? Well, that's something we all faced on Tuesday. You know, should we have red wine or white wine? Should we take the red or the blue pill? Should we push the person in the trolley problem, or should we just watch people die? Mark Twain made it pretty clear. I have a higher and grander standard of principle than George Washington or Martin Luther. We talked about him, right? He could not lie. I can, but I won't. Should we go around the big sequoia on the left or on the right? Should Buridan's donkey pick from one side or the other? Should we pick water or water? As Al so nicely showed us in the very beginning of this event, right? And this question becomes more tricky, of course, when it's water or water. But if someone tells you, okay, puts a gun to your head and says, okay, one of them has poison, you don't know which one. Is that then picking or choosing, right? This, in the original definition, if it's just water, it's clearly picking. And it's a fairly easy choice. It doesn't take a lot of deliberation. But once you know, and this is where information comes in, once you know that one of them contains poison, it's a completely different situation. So the environment is exactly the same, except for what you know about the environment. So we'll pick or choose some examples of where animals make choices that can be more or less related in the spectrum of different situations. And that's why I picked a lot of them with a lot of different kinds of connotations to see that, well, you know, maybe you can think of one of those examples that fit to those things that we're going to talk about now. The first thing we're going to look at, the first animal we're going to look at is a leech. And this one has recently eaten. It's quite fat and big. And what these animals do, they live in ponds usually, and you can find them around almost anywhere in Europe and in other places as well. And they feed on blood. And if you touch them a little bit here, let's say, touch them a little bit, they make a local bend, which means that they withdraw from that touch. They feel it quite well. If you touch them a little bit more, they will start crawling away if they get annoyed by what you're doing to them. And if you hit them really hard, they will start swim away because, no, I don't want to do that. So they can sense their environment just fine. And however, when they attach to your leg while you've been walking through, the animal's been foraging, it's been walking around, and it's noticed there's some movement in the water, it moves towards you, maybe smells you, attaches to your leg. Then, of course, while you're walking through that pond, it would be futile, it would be counterproductive that if you happen to brush by something, that the animal would start swimming away when it gets the exact same input that it would be getting when it's not feeding. So what's actually happening is that these animals stop responding to these stimuli. And in fact, when they're feeding, you can actually dissect them without anesthesia. So you can dissect the animal from the back, open it up, record from its nervous system while the animal is feeding, which you cannot do when the animal is not feeding. And so from these kinds of experiments, what you can do is there are preparations in neuroscience in many different animals that people call the Frankenstein preps, because you have an intact animal here in the front, and then you have the nervous system of the animal, the rest of the posterior part of the nervous system here in the back, and you have electrodes here, such that you can record from the nervous system. And this is a so-called local bend circuit. So you have the skin, you have tactile sensory neurons, the P neurons here, that then connect to the local bend interneurons that make connections to motor neurons and that then contract the muscles. And so, like I said, if you give via one electrode, you get always the same stimulation. You can either then trigger a local bend response or a crawl response or a swim response. And you can record these responses either in the intact animal, or you can do a intact part of the animal, or you can record it from the nervous system, because that's the response that the nervous system shows. And what happens when the animal is feeding, you can have the animal feed, and so you can do the same sort of experiments. And what people have found out is that serotonin then floods the nervous system when the animal is feeding. And what that serotonin does, it acts on receptors that the neurons have. And in the case of this experiment, what serotonin does, it doesn't do anything to those neurons themselves. They can still perceive the stimulus. They fire to the stimulus, but it's the vesicle release that happens at the synapses of those neurons that synapse onto the local bend interneurons. They don't release as much transmitter anymore as before, meaning that the sensory neurons are firing, but they're not communicating their activity to the rest of the nervous system. So the rest of the nervous system never gets the information that there is actually a sensory stimulus, even though the sensory neurons are perceiving it. And interestingly, to me, one could think the behavior initially looks like a choice, and that's what Quentin Godry and Bill Kristen called it. They called it behavioral choice, and this is from 2009, right? So this is quite some time ago. One of the first studies where you could really show that sort of thing. to me, what looks like a decision, and to the authors as well, doesn't really, when I see how the neurons are doing, it doesn't somehow feel like a decision anymore. It sort of looks more like an attention kind of process, where if I'm feeding, I don't want to attend to these other stimuli that may be distracting me from my feeding activity that I'm doing right now. Another example has to do with courtship. Although it will not be courtship in humans, it will be courtship in flies. And the way flies court, it's a quite regular stepwise process where the female decides if the next step happens. Or the female can decide if the next step happens. It doesn't have to, but so there's no rape in flies. So the female always is in control and decides what happens next. And the animal approaches the female and then starts orienting towards the front. It starts to tap the female, and then if the female doesn't run or fly away, it's taken, and other signals are used by the male as being that the female is being receptive. It will start to sing, it will extend the wing and it start to sing. It will follow the female and start licking its genitalia, will attempt copulation, and if the female then is ready, it will open its genital plates and the male will copulate with the female. And what you can do is, and this has been studied for a very long time, and the visual system of the flies have been studied for a very long time. And now we also have the connectome, meaning that all this work of the past decades about vision and courtship can be brought together by looking at the more central neurons, where these peripheral neurons that people have been studying before are now coming together. And so the visual system is very well studied because you can make animals, or these flies, you can make them escape. So people know precisely which kinds of stimuli can trigger which kind of escape. A very fast and abrupt escape for very strong stimuli, and then maybe a stopping, freezing, and running backwards escape, these sorts of things you can do. For instance, you can do this with a looming stimulus or with a moving shadow. And what will happen if you do this in this stage of the courtship, what happens is that the animal will stop courting and sometimes will just go away. But if you do it when the animal is really close, right? So imagine there's a big bully coming and you've laid your eyes on an attractive mate, possible mate, and that bully is sitting somewhere close. You may run away and say, it's not worth it. But, if it's already your second date, you're sitting close together and you're holding hands, and then the bully is coming, you may choose a different option. And so the closer you are to finalizing courtship, the less relevant this kind of threat gets. And so what you see, and you can see this in the behavior of the animals, is that the decision is being made that, well, I'd rather flee than court, and here I'd rather court than flee. And it seems on the outset as and I'm not pretending that the flies are making these computations, but from the outset, from looking at the situation, it seems like a very rational choice. If I'm very close to copulation, it's worth taking some risks, or it's worth taking more risks than if I'm really far away from copulation, I might find another mate. And so let's have a look at the different kinds of neurons or types of neurons that have to do with these different segments of this situation. So threat assessment is not being done in the optic lobes. So what you see here is a three-dimensional rendering of the fly brain. You have the antennal lobes here in front, which means that the animal is facing you. It's the anterior part of the animal. You have the optic lobes here where the eyes sit on the outside. And what you have is, and this is a schematic depiction, here you have a class of neurons, they're called the LC16 neurons. They sit downstream of the visual system, and they set the threat. There's different classes, that's why they're called LC16. There's many different classes of these LC neurons. And the job of these LC16 neurons is to tell the rest, or the downstream neurons, there is this particular type of threat. It's not so bad that you have to run away right away, but it's also not nothing, right? So you have to pay attention to this. It's sort of like one of the flies Jennifer Aniston cells if you want. It receives input from many other neurons that together specify this particular type of threat. Such that if you record from them, this is an imaging experiment, and if there's no looming stimulus, they show some baseline activity, and then they show ramped up activity when there's a threat, and they keep firing for some time after that. Now, I've said that it's conceivable that there's some ramping up that goes on during courtship as well. And there's a particular set of dopamine neurons that if you record from them during courtship, what you also find, and I've just widened this image up, if you record from them during courtship, also their activity ramps up. They get more and more active as courtship progresses over these, in this case, for instance, 4 minutes. So now what happens if you record from these LC16 neurons during the courtship, and you record instances where the threat comes early during courtship and when it comes late during courtship. And what you see is that if you have an early threat during courtship, and this is the 2-photon microscope with which these neurons get recorded, baseline, then comes the threat, and you see the threat neurons are firing. Whereas late during courtship, when the animal has already courted for several minutes and the female may look like it is perceptive, then these neurons stop firing. Now in this case, and this is a very recent paper, in this case the authors did not call it a decision. What they say is that this is a problem of love, or this is a process of love blindness, such that mating proximity blinds threat perception. Now it has to be emphasized that the neurons, the optical system, that the optical lobes, the animal is perceiving the moving stimulus. It's not that it's not seeing it. It's the assessment as a threat that is being changed by the proximity to mating. So again, what looks like a perfectly rational decision, well, if it's like this, I'm going to do that. If it's that, I'm going to do this, which looks like a decision-making algorithm, again, to me, looks more like, and to the authors here as well, looks more like a process of attention. If I'm really close to mating, I'm not going to attend to threatening stimuli. I see them, but I'm not going to classify them as threatening. And I think that tells us something about how evolution has shaped these kinds of circuits and how neurons are doing these sorts of things. Now, those were all examples that did not come from my lab. These were in both cases, friends and colleagues that I got to know over the years. But I picked these examples because I thought they're very powerful examples. And one of them was old, one of them was newer, just to see sort of what the kind of state of the art is right now, what the kind of things are that we can do and how we can start to explain how animals behave. And again, mammals are still far away from being able, or people who work in mammals are quite far away from doing that on that kind of level. But it's only a matter of time, I would say, until we reach something that's at least close. In our lab, we study, again, other kinds of decision-making for largely historical reasons, but also because I sometimes go to meetings like these and they give me ideas about experiments that maybe otherwise I wouldn't have done. And so I think here's an example about something that's fairly close to picking between red wine or white wine, at least if you have a very strong preference for one over the other, but not an exclusionary preference. And so what we have, the experiment I want to talk about in the next couple of slides, is one where you first put animals in a tube like this, and you let them rest there for a while, and then it's hard to see with the light, but there's a movable part in the middle that contains a hole, which means that we can tap the flies into this so-called elevator. You pull this up, there's an elevator hole here, you tap the flies in there, push them to the bottom, and then you give them a couple of seconds, half a minute, a minute. You give them time to choose. There's light on top here. Give them time to choose the bright arm over the dark arm. And we've designed this, and this actually, curiously enough, has to do with the size, the length of these tubes, believe it or not, for some reasons. Flies really, for flies, the length of these tubes really matters, and we've adjusted the size of these tubes such that we would get about 30% of the animals being either in the dark arm or caught in the elevator when we pull it up to count the flies, and 70% are then in the bright arm, which is something that you'd expect. You know, insects, as we all know, move towards the light. It's called phototaxis. In this case, they can't move towards the light because the light is on top. They prefer either the bright arm or the dark arm. So this is not phototaxis, this is photopreference. And you could ask yourself, okay, I thought flies, you know, insects always go to the light. What's wrong with those 30% that are not in the light? And there's an easy experiment to do. Well, you take those 70%, you test them again, and what you find is, oh, they all again split up 70-30, and you take those 30%, and what you find is, again, they split up 70-30. So what that shows, and there's work by others that essentially shows this 70% that we have here is actually not that each fly has a 70% preference. It's actually an average of averages. So if you test each fly many, many times, you get an average of preference. And then if you average many, many flies, you get 70%. So you have some that are 90% towards the light, some that are 10% towards the light. Most of them are somewhere in this 70, 80, 60% range. So this means that, and these sorts of experiments, this is again just an example of many types of experiments that gave rise around the 80s to the Harvard Law of Animal Behavior that says that under carefully controlled experimental circumstances, an animal will behave as it damn well pleases. And shows that even under those circumstances, there's some fundamental unpredictability that's in there. We cannot tell. Even in genetically identical animals, they develop these different preferences. And so you cannot tell when the first time the animal is in there if it's going to be one of those animals that goes to the light arm or one of those that goes to the dark arm. And moreover, one can show that this is not just something that happens to be there. It's actually the case that the flies makes some kinds of evaluations before they then choose whether they go to the light, bright, or the dark arm. So another way of calculating where the flies are going is to count all the flies, take bright minus dark, and divide it by the total. Then you get a score that goes from -1 to 1. When the flies are divided 50-50, it's a 0. If they go all to the dark, it's -1. If they go all to the bright arm, it's +1. And if they're around 50, then you have 75% of the animals going to the bright arm. And this is like, you know, just under there. So we're around about those 70% in these flies. And then a heroic postdoc in our lab a couple of years ago, Axel Godostica, then took sugar water to glue the wings of the fly so that they couldn't fly anymore. And he did all kinds of other ways to manipulate flight behavior. This is just one example of many where we just use different methods to make the flies not able to fly anymore. And what then happens is that the flies go away from the light. And then you could think that could be all kinds of things, right? We could have damaged the flies while we were doing this. And so you douse them in distilled water that dissolves the sugar. You let them dry for a while, and we put them back in, and they go again to the light. So, long story short, we tested many other things. The one conclusion out of these types of experiments is that before flies make a decision whether they go to the bright arm or to the dark arm, is that they check, can I fly or not? Or they've checked it long before, and they know they can fly or not. And then when they have a decision to make between a bright and a dark arm, they either chose the bright arm if they can fly or the dark arm if they can't. But again, it's not an all or nothing decision. It's a preference. And that preference actually can also be shifted, interestingly, by neurons that are known to be involved in valuation. So octopamine and dopamine are substances, or octopaminergic and dopaminergic neurons, I should more correctly say, that are known to be involved in valuation. In learning and memory, you can activate these kinds of neurons and then pair the activation of such neurons with, let's say, an odor, and that odor becomes appetitive or it becomes aversive, right? So these types of neurons have been shown to be involved in valuation processes. And now come two slides very quickly that may look, that are a lot of data, that are a lot of work, but a lot of it, 2/3 of this is controls. That's a nice thing of working in flies. You can do a lot of all, you can essentially do all, almost always, all do the necessary controls that you have to do. So this time we didn't glue the wings, we just cut the wings, and everything that you see here is control flies for these experimental flies. So I don't have time to go into the genetic details, but those are all controls for various factors, both genetic and handling factors. And what they show is that if the wings are intact, all the control flies go to the light as they should. And in this case, if you cut the wings, all the control flies go away from the light as they should, or well, you know, the median goes away from the light. So the controls show the behavior that the wild-type flies do. And then in this case, what we do is in green, we have the same situation as before, with the same situation as before, where we have the flies can fly, the flies cannot fly, and the flies can fly. This is what we did here with the glue. In this case, it's again a similar aspect, but we do it it's a different design of the experiment. So we have room temperature, then we have 32 degrees, which for the flies is barely okay. And then again, we have again the room temperature. And what we do with this change in temperature is that we switch off the neurons that are either dopaminergic or octopaminergic, right? And we do this either in flies that can fly or in flies that cannot fly. So let's look at dopamine. At room temperature, when dopamine is active, the flies with wings go to the light. This positive photopreference is gone when dopamine is blocked and comes back when dopamine is active. Dopaminergic neurons are active. In clipped wings flies, this has no effect. In octopamine, it looks the other way around. If we block octopamine, there's no effect on flies with intact wings. They go to the light, but their negative photopreference is abolished when you block octopaminergic neurons. And again, the same thing, we can use temperature to activate neurons. And again, here the controls show everything is perfectly fine. Let's look at dopamine. If we activate dopaminergic neurons in flies that are already going to the light, it has no effect. But if we activate dopamine in flies that have clipped wings, we can actually make them behave as if they had intact wings. So we can shift their preference from away to the light to the light. And it's the inverse for octopamine. Flies that have intact wings that would usually go towards the light. If you activate octopaminergic neurons, that positive photopreference is gone. And we have no effect on negative photopreference. So this is, you know, a very simple experiment in very comparatively simple animals. And you already there see that this is not a decision that's an all or nothing decision. It's a preference. That preference is not all or none. It's a probabilistic preference that tells me I have a 70% preference for this or for that. And we are starting to find out how the nervous system, we're not at that level yet as the previous examples, but we're starting to find out how the nervous system is doing that sort of thing. But it looks to me, from what we found, that this is a very fundamentally different kind of decision, even though also dopamine is involved in this case, but in a seemingly quite different kind of way. Now, and yet another different kind of decisions that I would say would be very similar to, in a conceptual way, similar to Buridan's donkey with two identical haystacks, is by simply taking away all the environment and not giving the animal anything to choose from. And we do this in stationary flying flies, where the flies are glued here by head and thorax to a hook. That hook is then used to tether the fly, and the fly can, it can move its antennae, it can beat its wings, it could move its abdomen and legs and that sort of thing. But the only thing that we're actually looking at, and this is an old depiction of an old torque meter, we have newer ones, this then measures the tendency of the fly to perform left or right turns around its vertical body axis. In this time-lapse video, you see in 30 seconds a 30-minute time trace that looks like this. And even if you place the animal in a completely featureless environment, the animals still change their direction, or they attempted turning direction from, let's say in this case, from turning mostly left to turning mostly right. And there's nothing in the environment that tells the animal to do that, right? And people have studied these tethered flies for decades, long before I was born. So we know very well which kinds of stimuli make the animal go to the left and to the right, and we can very well control the situation and say none of those stimuli that usually pull the fly in one or the other direction are now present. Now this is obviously, this is not as clean as just taking the nervous system out and just cutting all sensory organs off, but it's sort of the next best way that you can still look at intact behavior, but still isolate it as much as possible from potential stimuli. So I would say, and this is something that probably comes more from the German language, where action and reaction can be perceived also from the Latin perspective as antonyms, right? Action is something that happens out of your own body, whereas reaction, as it would imply, is always something that you do after something else has happened, right? So we would call this actions and not reactions or responses. And so the question now is, okay, if there is this variability, what can we get from that? So how can we get to the nature of these decisions when there's nothing in the environment that tells the animal where to go? And one hypothesis could be that these animals operate like analog radios. You know, you take the antenna off and what you get out is just pure noise. And one way to test this is that you can look at the behavior that the animals do and analyze it mathematically. And we did all kinds of analyses that I don't have time to go into. And one of the last analyses we did was to distinguish the behavior of the animals, to distinguish it from nonlinear behavior. Also, we heard from Henry's talk that nonlinearities can sometimes, or nonlinear systems can sometimes behave unpredictably, and can therefore look to the human eye, they can look very stochastic. But there are mathematical methods with which you can actually distinguish them from something that's actually stochastic. And one way of doing this is a nonlinear forecasting procedure, where you take some portion of the data that you have, let's say whatever, 15 minutes of those 30 minutes that we have, you derive a computer model, sort of like a weather forecasting model, and then you predict the second half and try to see how good is that prediction, because I can take that weather model and make a linear prediction that would be random or a linear relationship, or I can tune non-linear, and so I can quantify, you know, how linear, how non-linear is this, is the data or the signature that I can find in the data, right? So we can tune the model from being linear to be most non-linear, and then we compare the prediction with the actual data, and we say that the better the model predicts the actual data, the more confident we would be that the outcome is actually somehow related to the way the model functions. And so what you would expect if you have a linear or a stochastic time series as an input is you would get a flat line here, because if it's linear, there's no point in making a nonlinear prediction. I can tune this to be more nonlinear, I don't get any better. However, if I have a nonlinear time series or nonlinear mechanism underlying the time series, what I would expect is I would, in principle, also get a higher, depending on the linearity, but if this were a stochastic time series, I would expect a higher predictability by the model, generally, and then one that would increase with increased nonlinearity of the computer model. And if we take flies, this is exactly what we find. We find that the more nonlinear I make the prediction, the better the predictability of the flies gets. Meaning that the outcome of, or the output of the flies that they do is not just random noise, it's something that is nonlinear, something that we are all very familiar with. What I didn't know when we implemented this method is, like, what exactly is this method really detecting? And so what we did, and what's a standard procedure if you use computational methods to study real-world data, is that you give it data where you know what you put in, right? You don't just use it as a black box, you test it. So you have a positive control and a negative control, and we used a computer model from the literature to generate different types of nonlinearities. And so we could get this agent to generate data that looked even more nonlinear than the fly data, but we could also make it tune it such that the computer model would be like a linear model. Now, interestingly, and I don't have time to go into these different traces. Interestingly, this software agent runs on a very famous equation. It's a logistic map. And very famously, and this harks back to Henry's concept of a fuzzy future rather than a forking path. If you look at this equation, it just exists, it just contains one parameter, that's r. The rest is just that the state of the system at the time point n+1 is dependent on the state of the system at the time point n, and this parameter. And so if you keep iterating this, let's say for 10,000 times, and you set the parameter to 2.4, it converges on something that's just below 0.6. However, if you tune r to be larger and larger until it hits 4, what you find is that you get this very famous Feigenbaum diagram, where things get really, really fuzzy here at the end. And the important point that I want to make here for this particular type of experiments is that this nonlinear forecasting procedure provides you with a straight stochastic line for this part of the graph of R. But if we tune R in this area, this is when we get this steep increase. This means that this is a nonlinear function, but it behaves linearly. So the behavior of the flies is somehow more similar, mathematically, more similar to this part of the graph than to this part of the graph. So it's not just any type of behavior that this procedure is detecting, it's detecting this kind of what people call mathematically unstable nonlinearity. Now, the final example that I want to talk about is the marine snail aplysia. And what you will see here is that this is the mouth of the animal, those are the lips, and in here you see the jaws. And I've been mean to this animal because I've starved it for some time. And then what the animals do is that they look for food. And because I wanted to make a video with a couple of bites on it, I squeezed some sushi seaweed. I soaked it in water and I squeezed it in there so that the water would contain smell of seaweed, but nothing else. So the animal is hungry and it smells like seaweed. What that does is it increases the probability of the animal to produce feeding behavior, even though there's no food there other than the smell. So there's no mechanical stimulus there that would tell the animal, oh, this is seaweed, you should eat this. It's just that the animal is hungry, it smells like seaweed, so it's going to start to bite. And what you will see happening is that a tongue-like organ called the radula, it has two halves, and it usually sits in the back of the head. It gets protracted, it tries to grab something, and then gets retracted to grab that food that the animal, I would presume, hopes is there, gets into the stomach of the animal. So this is what we need to do, right? There you go. Oh, come on. No. That is weird. Usually this works. This is a video I've used many times, but we can also do this differently. This has to be that way. That is weird, why this is not working. So, is that one that I have? Well, that's a different type, different animal, but it's the same behavior. See, the radula comes out and goes back. Now, this is an older video with less resolution, but you get the idea. I can run it again. And in the video that didn't run, it would just do 3 of those in a row. And in different places. So animals do this type of behavior even in the absence of the seaweed smell, but really, really rarely. And they basically never do it when they're not hungry. And what you can do, you can take a, let's see, does it run now? Yeah, there you go. Don't ask me why it's working. You can see different, right? You can see the different bites. There comes another one. Regula comes out, tries to grab something, and goes back. So this works. Animals do this sort of thing all the time. Now, what you can also do is you can take the ganglia. So these snails have a distributed nervous system. You can take the ganglia that control this behavior. You can take them out, put them in a dish, similar to the leech nervous system, put electrodes on there and record from them. So what you then see, you can take the protraction nerve, the retraction nerve, and the closure nerve, and you see, and we'll just look on this part for now, that we'll leave this one out. You have firing in the protraction, you have firing in the retraction, and then the closure happens during the retraction, right? So this is what you get. And you get that even in the dish. So even if you cut off all the sensory organs, the ganglia do this on their own. Now, we know in these animals, we know the different neurons that are in these ganglia and what they do. And so I'm not going to go into what it looks like. They have different names. They're called B for buccal, and they have different numbers. The important part that I want to emphasize here is you look at those nerve recordings, protraction and retraction. We just look at protraction and retraction, and you find neurons like these here that fire before the behavior actually happens. You have many other neurons that do other things, right? But those are the important ones. There are 3 neurons, each one of them fires before, and some of those, fire before the other ones. Any one of those, if you put an electrode in them and you fire them, the behavior starts. Any 3 of them, doesn't matter which one. And those behaviors that I showed in those 2 different animals, the 2 different videos, they all look the same, right? The radula comes out, gets grabbed, goes in. Looks like it's always the same. Also here you see protraction, retraction, right? Protraction, retraction, protraction, retraction. It's always pretty much the same. But it's not. It looks deterministic, but it's actually highly variable when you look at it on the neuronal perspective. So here you have these 3 neurons again, B63, B65, and B30. And now we look at, Romuald Nargeot, a colleague in Bordeaux, looked at which neuron fires first. And here, B65 fires first, then comes B63, and then B30. The next one around, so those are all recordings that are happening at the same time, right? Same animal, same preparation, same time. In this pattern, it's 1, 2, and 3. In this pattern, 1, 2, 3. In this pattern, 3, 2, 1, and so on. Every single time, what looks to us to be the same behavior is actually coordinated by a totally different set of neurons that kick the behavior off. So how can that happen, right? So I'm very happy that Peter introduced this concept of threshold, so I don't have to do that anymore. You put an electrode into a cell, you give it hyperpolarizing or depolarizing input, and then what you find is that, the more depolarizing input you put in there, the more action potentials you get. This is just a very deterministic neuron because it's a computer neuron. This is not a real neuron, it's a textbook neuron. We don't really know how many neurons actually behave like that, given that there's thousands and thousands of different types of neurons. But it's very often, we teach our students that this is the neuron, which is not the case. Because, for instance, there's also neurons like the B63 neuron. This B63 neuron, if you isolate it completely from all the other neurons in the ganglion, you depolarize it just a little bit, and it bursts and then stops bursting. Bursts, stops bursting, goes up and down, up and down. How does it do that? So it behaves dramatically, radically different from any of the types of neurons you would find in a review article. You usually don't find them in textbooks, but in a review article you can find different types of neurons described. This behaves radically different. And quite recently, they have found, Romuald Nargeot and colleagues have found out that it's a molecular process that starts to look, it's not completely elucidated yet, it starts to look like a double pendulum. And here you see how chaotic a double pendulum swings. It's a classic model of a nonlinear system. And if you want, the first arm of the pendulum is endoplasmic calcium that oscillates from intercellular stores into the cell and out again. And that interacts then with extracellular calcium, extracellular calcium, and neuronal activity, so other conductances in the cell, such that the total membrane potential, so if this is potential that's generated by the internal calcium, then the total membrane potential then oscillates chaotically because of those interactions. So it looks as if, and we can get into the neuroethology of this, it looks as if this neuron evolved precisely to not behave deterministically. Precisely, it evolved to throw dice. It evolved to do things spontaneously in a situation that's underdetermined. Right? So this is how you get nonlinear chaotic dynamics, and you get a neuron to fire spontaneously when all you need is a little bit of depolarization. When the animal is hungry, when the animal smells seaweed, maybe then it will spontaneously sometimes start to bite and sometimes not. So those are all examples, especially the last ones that I showed, that where the situation was explicitly and experimentally made as underdetermined as we possibly could. And in lots of situations, this is how animals evolved their capabilities of doing things, of acting to survive. Because in many, many situations, the genes and then the environment and lifestyle and past that we had are not sufficient to tell the individual what to do. It's not sufficient. There has to be some way in order to be able to do something when I don't have enough instruction, what exactly to do. So this is what I think underdetermined means. This is why I think the concept that Henry proposed of the world being underdetermined, why this I think is a useful concept and why it can help us understand sometimes types of decisions, and while other types of decisions may look like decision-making happens actually in a very, very different way. And so with that, I would like to thank you.